Hello everyone and welcome to another episode of Nowhere Yudarris. This is your host Mahmoud Nowhere. So we're going to do a little bit of carbohydrate structure and function in biochemistry today. We'll cover the structure of carbohydrates, what they're made of, etc, etc. So shall we start? Don't forget to like, share, and subscribe, and leave any comments of what topics you want me to cover next. Carbohydrates are basically sugars with monosaccharides. They are made of monosaccharide units. And glycerolaldehydes are aldehyde sugars linked with glycosidic linkages. So glycerolaldehydes, aldehyde sugars, so aldehyde sugars linked with glycosidic linkages. Some common sugars include glucose, fructose, lactose, and mannose. These are some common carbohydrate sugars. So enantiomers, I covered this in a isomers video. These are basically super non-imposable images of two stereoisomers. That means that I want you, for instance, to um, imagine like a hand in front of a mirror, and then when you put this hand, when you take both hands, you see how, for instance, they don't add up to each other. So you have this, and then you have this. That's what super non-imposable means. Due to the increased number of chiral centers. So enantiomers, when you have chiral centers. So you have two types. You have D stereoisomer images and L stereoisomer images. So with the D, in order to differentiate between them, you need to look at the um, carbohydrate. So if the hydroxides are on the right side, or the highest chiral center is on the right side, like we see here, you see I have 1, 2, 3, 4 chiral centers. So basically you have 1, 2, 3 on the right side. That's a D carb. Alright, the stereoisomer. But if it's on the left, you have 3 here on the left. That's a L stereoisomer. Alright, and the same, these are enantiomers. Alright, enantiomers, they're super non-imposable images, but they're the same structure. They're just a different orientation. And here's the same thing. You see, you have one chiral center here, and the main group is on the left side. But here, the dealanine, the main group, is on the right side. That's how you determine the absolute configuration. Yep. And then we said the same sugar are different optical families, and they are enantiomers. Since monosaccharide contains both hydroxyl groups, they can undergo a intramolecular reaction to form cyclic hemiacyls or hemikettles. So basically, you see how a carbohydrate has a hydroxyl group. So using two hydroxyl groups, they can turn into a cyclic structure using intramolecular reactions. So a hemikettle is when a hemikettle forms, that forms a ketone, alright? But when a hemiacetal forms, that's an aldehyde. Monosaccharides oxidize to yield energy. Aldehydes oxidize to become carboxylic acid. So you can turn, or you can generate a carboxylic acid from an aldehyde. And oxidized aldoses are reducing agents. Tolins and Benedicts are two tests and two reagents used to detect reducing sugar. As you can see here, Tolin's reagent is silver NH3 and it's reduced to produce silvery mirror when aldehyde is present. So it reacts with an aldose or aldehyde in order to detect it, and a positive detection yields silver or silvery mirror. And then Benedict uses copper 2 citrate or copper 2 in order to detect detect for glucose as well. And the Benedict reagent itself, the the copper citrate 2 is blue, but when it's a positive detection, it turns into a red precipitation. And then sugars react with carboxylic acid in order to form esters. So to form a carboxylic acid, you oxidize aldehyde, but what if you want to form an ester? To form an ester, sugars react with carboxylic acids. Disaccharides is basically when you have two monosaccharides joined together, such as sucrose, lactose, and maltose. And then you have homopolysaccharides, and these compose of glucose, such as cellulose of plant. So homopolysaccharides are basically kind of the same monomer, and like same type of unit, but with then on the other side you have heteropolysaccharide composed of more than one type of monosaccharide, more than one type of monomer, a mix basically. And then lastly you have glycogen, which functions as the main energy storage for animals. Humans also have glycogen stored, and then that's it. That's a wrap up. It's a quick video. Make sure to like, subscribe, and leave any comments, and let me know if there's anything else I can explain later on.